Hello everybody, you are welcome. Today we will answer together variant 41 October November 24. Let's start. This figure 1.1 is photo micrograph of cross section of part of a leaf with certain letters K, L, M, N, J. The question here, state letter A single I states the letter in figure 1.1 that identifies where gas exchange is controlled. As we know, gas exchange takes place through certain opening called stomata. Stomata found in most normal leaves in the lower epidermis. So look at layer L, okay, to see guard cells that control this stoma or stomata. Okay, look here, this stoma, another one like this in lower epidermis, found in lower epidermis, mostly found in lower epidermis. Most photosynthesis occurs. Photosynthesis takes place in mesophyll layer. Mesophyll layer divided into two parts, N and M. In palisade, where most photosynthesis occurs, while spongy chair or helps photosynthesis, but the main function of spongy mesophyll cells for gaseous exchange, okay, because a lot of air space is found in between. Look here, N, palisade mesophyll, N. Spongy mesophyll cells are found where? In layer M, okay, layer M. So M, spongy mesophyll. Okay. Now draw a circle. Draw a circle on figure 1.1 to identify vascular bundle. Just drawing a circle around a vascular bundle. Where is a vascular bundle found here? Look at this part. Vascular bundle where xylem and phloem tissues as conductive tissues found inside this bundle, okay, for transportation. Xylem for water and minerals, and the phloem for ready-made photosynthesizing material, I mean sucrose and amino acids, okay, to all part of the plant. Now letter triple I, translocation occurs in the vascular bundle. States the names of two substances that are only moved by translocation. Only moved by translocation. Translocation means assimilates or products of photosynthesis like sucrose, and also later on the plant leaf cells make certain important material called amino acids, are translocated to another part of the plant where they are needed or stored. Here look for 2 marks, sucrose and amino acids, okay. Here in this figure, the source, the photosynthesizing leaves, green leaves, make glucose in photosynthesis, converted into sucrose to be transported, okay, plus it form also amino acids as raw material for protein, and sucrose for carbohydrate are transported from source, okay, I mean the leaves, into all parts of the plant through phloem, okay, where it may be stored in sink or reach fruit, flower, root hair cells, where they are used for metabolism, okay, or for storage. Now letter B, explain how xylem vessels are adapted for their functions. How xylem vessels are adapted for their functions. This LS, longitudinal section, and transverse section TS, okay, showing xylem vessel. This certain photomicrograph, okay, taken by certain type of microscope to show the structure of xylem vessels. Here where lignification or lignin deposit in certain shapes inside xylem vessels to support the xylem vessels and prevent collapse during transportation of water and the mineral inside. Okay, look, number one, thick walls containing lignin for support. Thick walls containing lignin for support. This is the first adaptation. Second one, no cell contents, where all cells are died and all protoplasm, I mean cytoplasm and the nucleus, are decayed. Okay, decayed. At early stage of xylem cells, they were living, but later on, to be adapted for transportation, for transport, it must be hollow. So no cell contents, I mean hollow lumen, for uninterrupted water flow. Uninterrupted water flow. Cells are joined end to end to form continuous tubes, long continuous tubes. Narrow diameter, narrow lumen to prevent water column breaking. Okay, narrow diameter to help capillary action like straw when we put it inside juice. Okay, here pets and the walls allow sideways. Look at this pets here and here. Okay, this pets in the walls allow sideways movement of water during movement of water inside the lumen. Okay, it moves out of this xylem vessels. Now letter C. Describe how water moves through a plant from the soil to the air spaces in a leaf. How many marks? 4 marks for this question. Look at this figure. And water, firstly, water enters root hair cells by osmosis from high water potential in soil to low water potential in the root hair cell. Okay, it moves by osmosis from high to low down its water potential gradient. It's okay. That's number one. Second statement here, water moves through root cortex, all this part of section, the cells of cortex, all of these layers. Okay, then it moves by osmosis also, via osmosis to the xylem. Now it reaches xylem vessels. Okay, water travels up the xylem vessels as continuous column without breaking due to cohesion. Cohesion means water molecules joined together by hydrogen bonds. Attraction between water molecules. Here look, water move through xylem vessels, continuous column, due to cohesion and adhesion forces of water molecules. Cohesion due to attraction forces between water molecules in the form of hydrogen bonds, and adhesion means sticking water molecules to the wall of xylem vessels. Okay, now water moves out of xylem into mesophyll cells. Okay, water evaporates from mesophyll cell surfaces into the air. It moves out and towards all cells of mesophyll layer, palisade and spongy. Okay, plus water moved outside the plant, okay, in the form of water vapor. This is called transpiration. Okay, by diffusion, water vapor exit stomata by diffusion in the form of gas, water vapor. Thus explaining how water moves through plant from the soil till reaching air spaces in a leaf. Okay, now letter D. Explain why plants need nitrate ions. Nitrate ions in chemistry, NO3-1. Why plant needs nitrate ions? Nitrate ions, NO3-1, are used to make amino acids, which are the building bricks for proteins. Okay, look at the structure of amino acids here where N is found. Okay, as you know, all protein containing four elements C, H, O, N. C, H, O, and N. N is essential where it is needed to make or build up amino acids, different amino acids. Amino acids, which are the building blocks of proteins. All proteins are made of sequence of amino acids. Proteins are essential for plant growth, enzyme production. All enzymes are protein, and structural components. Some structural components in the plant tissues. It's okay. Here look at the protein chain as polypeptide chain, sequence of amino acids, each amino acid containing N. How the plant obtain nitrogen, not from the air like N2, but plant obtains the nitrogen in the form of nitrates. Refer to nitrogen cycle at the end of your course. Okay. Second question. Chlamydia. Chlamydia is a sexually transmitted infection, STI, sexually transmitted infection, that is caused by bacterium. Figure 2.1 shows the number of chlamydia cases in a country in 2018. In certain country, this statistical study about chlamydia cases in this country. Look at the x-axis, age group per years, and y-axis as dependent variable, cases per 100,000 people. Okay, now look for this part, letter single I, A single I, calculate the percentage decrease in the number of chlamydia cases between the age groups 20 up to 24 and 25 up to 29. Give your answer to two significant figures. Space for working. Okay, now look at this age group from 2024. It creates number of cases equal what? Equal 3,900. Okay, while in the second group, next group, starting from 25 years up to 29, how many cases? 1,600. Here he asked me about calculation of the percentage decrease. Percentage decrease or percentage increase calculated by the same rule. Final minus initial, divide initial times 100. Final minus initial, divide initial times 100. Look, where is final? Final is the second group. Okay, look, 1,600 minus 3,900, divide initial 3,900 times 100. Okay, to create percentage. By calculation, by calculator, you find negative sign. This showing you are correct. Here you ask about percentage decrease. So the decrease always starting with minus sign. Here minus 58.97%. But look, again go back to give your answer to two significant figures. I have to approximate this percentage into two significant figures. How come? Look, minus 59%. So the cases decreases by about 59% from this first group to the second group. Okay, Now describe, letter double I, describe how the spread of sexually transmitted infections such as chlamydia can be controlled. In our course, we discussed the spread of HIV infection that leads to AIDS. The same answer like AIDS, the same answer like the lesson AIDS. Okay, look how the spread of sexually transmitted infection such as chlamydia can be controlled. Controlled how come? Abstain from sexual contact. Abstain from sexual contact. Number 2, use condoms or femidoms as barrier contraception barriers. For male, condom. For female, femidom. Okay, male condom put over the penis. Here, female condom put inside vagina to reach the area of cervix. Prevent reach of sperms or prevent mixing fluids between the two partners. Okay, here regular testing screening to identify infected individuals. Look at this rapid test to screen which one positive or negative. Here where sample put drops of sample like urine, like blood, like semen, like vaginal secretions put inside this hole. Okay, what happened in this test? 2 areas, okay, colored with red. This indicate positive result. Here for test, here for control to show validity of the test. It is valid. Okay, while here for testing the sample put here. Okay, regular testing screening to identify infected individuals. What about contact tracing to inform and test sexual partners? Contact tracing, I mean here if the husband is infected with chlamydia, not only the husband given the treatment or antibiotic to kill chlamydia bacteria, but also his partner, his wife, okay, or his partner. Contact tracing to inform and test sexual partners. Public education about transmission and prevention put inside courses or syllabuses or syllabi of certain boards like Cambridge, for example. Public education about transmission and prevention. Use of antibiotics to treat infections, to kill bacteria, if that's given by chlamydia, or for HIV is given certain drugs, okay, like ART, okay, antiretrovirus treatment if viral infection takes place by HIV. No sharing of needles, no sharing of needles to prevent cross infection, okay, during heroin abuse or drug abuse. Sharing needles, one of the reasons to transmit these infections, viral or bacterial infections. Screen the blood used for transfusion during blood donation, okay, to prevent transmission from infected person to another healthy person. Look, vaccination if available for sexually transmitted infection, if available. Look at vaccination, this method of controlling spread of diseases like bacterial or viral disease, some bacterial and viral diseases. It's okay. Now, chlamydia can damage the reproductive system. See the name of the part of the female reproductive system that produces estrogen. Estrogen, female hormone. It produced by, secreted by the ovary. Look, female sex hormones, estrogen and progesterone, secreted by the ovaries of the female. Estrogen, sometimes called estradiol as chemical name, okay, responsible for what? Sex specific changes at puberty, I mean secondary sexual characteristics, okay. Also both estrogen and progesterone responsible for monthly ovulation and menstruation. Okay, monthly ovulation and menstruation during the cycle. Look now, describes the role of estrogen at puberty. What its role? Okay, here development of secondary sexual characteristics under the control of pituitary gland. Certain message of pituitary gland, certain hormones reach the ovary to encourage ovary to produce estrogen. Estrogen now responsible for development of secondary sexual characteristics, initiates menstrual cycle, and regulates female fertility and ovulation also in the mid-cycle for regular cycle. Here hips of the female wedding become more wider, breast development, growth of pubic and body hair. All these features considered secondary sexual characteristics due to female hormone estrogen. Okay, now complete the sentences about drug resistance and genetic variation by writing suitable words or phrase in the spaces provided. Bacteria can be killed by drugs called, yes, called what? Antibiotics. Antibiotics are specific to kill bacteria by different mechanisms. Some affect cell wall building, others affect cell membrane synthesis. Some affect metabolism of bacteria. Some inhibit replication of DNA. Some inhibit protein synthesis, bind with ribosomes with different mechanisms. The development of strains of bacteria that are resistant to these drugs is an example of, yes, natural selection. Natural selection, this development of strains of antibiotic resistance bacteria as strong example, good example of natural selection, where nature select the fittest individuals that carry the genes for producing certain chemical to resist the action of antibiotics given during infection. Okay, to resist this action. Resistant strains of bacteria have gene mutations that enable them to survive drug treatment. Gene mutations are caused by random changes in space of bases in DNA. Here, sequence of bases A, G, C, T, bases or nucleotides in DNA, and result in the formation of new allele in the bacteria, different form of the same gene due to mutation, due to change in sequence of bases. Mutation rates can be increased by space radiation and some chemicals. When someone exposed to large dose of X-ray, gamma rays, ultraviolet, mutations increase in its percentage. Okay, mutation rates can be increased by ionizing radiation like X-rays, gamma rays, and so on. In animals and the plants, another source of genetic variation is a type of nuclear division called, yes, meiosis. Meiosis is responsible for genetic variation in sexual reproduction, where crossing over takes place, random assortment of chromosomes during meiosis takes place, all of this responsible for production of different types of gametes during meiosis. Okay. Question number 3. Letter A. Figure 3.1 shows the structure of pathogen that causes cholera. Okay, this pathogen causes cholera. What the question here states the name of kingdom that includes the pathogen shown in figure 3.1, just the name of the kingdom. As you know, cholera is bacterium. bacteria belong to kingdom prokaryotes. So the kingdom prokaryote, okay, as it lacks nucleus. As it lacks nucleus, no nucleus, I mean prokaryotic or prokaryote as kingdom. Here, table 3.1 shows some of the names, functions, or uses and identifying letters of the parts labeled in figure 3.1. Complete table 3.1. Look as bacterium, okay. For flagellum, the name flagellum, letter B, yes, letter B from the figure. It's the bacterium uses it for movement, okay. As we know, this bacteria spread or transmitted from contaminated water by sewage, from contaminated water to healthy individual to cause infection. It uses flagellum. Letter G, this the innermost line surrounding cytoplasm directly is cell membrane. Letter G, cell membrane. What is function? As you know, cell membrane is partially or semi-permeable membrane, allow exit and entry of material or substances needed for transportation or transport within the cell. Which letter responsible for protein synthesis? Letter D. It is called ribosome. Ribosomes, where translation of messenger RNA takes place to form polypeptide chains or protein. Cell wall. Here letter A represents cell wall, okay. Cell wall, its function prevents bursting and provides support of bacterial cell. Support and prevent bursting by preventing excess water entry to the bacterium, okay. Is letter A. Which letter or organelle or structure used by humans in genetic modification? Circular DNA plasmids. Letter C, okay, letter C. Letter B, describe how a person becomes infected with cholera. Look, with drinking contaminated water or eating contaminated food by this type of bacteria, okay. Drinking contaminated water or consuming contaminated food with this bacterium. Letter C, people with cholera can become very dehydrated. Explain how cholera causes dehydration. Look at this figure. This showing the infection with bacteria, okay. The bacteria passes from stomach to small intestine, okay. Here where bacteria found, they multiply, increase in number inside the lumen and secrete toxins. This toxin in green color adhere or stick with the microvilli or extensions of the epithelial cells of the gut, okay, and causes changes in the gates inside this cell surface membrane. It causes leakage of chloride ions from inside the intestinal cells to the lumen. Chloride ions exit to the lumen, followed by exit of water also. Water also diffuses or move out inside the lumen, causes watery feces. Why? Because when chloride ion exit, it makes or responsible for lowering water potential, while water inside epithelial cells with high water potential. So water with high water potential move out of the cell by osmosis to the lumen, causes watery feces. These steps must be written in your answer. Look, cholera bacteria produce toxins that disrupt intestinal function. Number 2, toxin causes second statement, toxins causes chloride ions to be secreted into the intestinal lumen. This lowers the water potential in the lumen, okay, and water moves out of intestinal cells into the gut and compensated by water and chloride ions from blood supply that enrich the intestinal cells. Okay, this leads to diarrhea and excessive water loss plus ions. Okay, results in severe dehydration. Okay, now for letter D, vaccination can help to prevent the spread of diseases such as cholera. Outline the process of vaccination and explain how it can prevent the spread of diseases. Process of vaccination. Vaccination provide the people with long-term immunity. How come? A vaccine may contain weakened or dead pathogens or the antigens of the pathogen. Okay, mainly the vaccine may be weakened, attenuated pathogen, dead pathogen, or their antigens found on the surface. Vaccination stimulates the immune system to what? To produce an immune response. How come? Mass vaccination programs reduce person-to-person transmission. Okay, this mass vaccination, most of the people of the country, okay, should have this vaccine, okay, to eradicate the spread of certain infectious disease like cholera or TB, for example. Look here, COVID-19. Here, vaccination center where people, okay, reach the center to be given this vaccine against COVID-19. The vaccine triggers white blood cells, I mean lymphocytes, to produce antibodies. These lymphocytes triggered by take-in vaccination, okay, the white blood cells from the type lymphocyte, B lymphocyte, okay, produces stimulated to divide, and some of this white blood cell, okay, produce antibodies. Memory cells are formed also for long-term immunity. Look at memory cells here. When lymphocyte subjected or reach with antigens on pathogen, specific antigen on specific pathogen, and complementary fat with its receptors, the cells of lymphocytes become activated and start to divide mitotically by mitosis into clone of cells. Okay, clone of cells. This clone of cells produced by mitosis and differentiate into two groups. Certain group waiting for second exposure of the same pathogen, okay, if real pathogen enter our body, okay, like this vaccine, okay, so memory cells are produced. The second group of cells start to produce and secrete antibodies in our bloodstream. This for clumping pathogen. This first exposure, second exposure done by memory cells. Look, memory cells are formed for long-term immunity. If the person is later exposed to the same pathogen, antibodies are produced rapidly with higher concentration. Look, first exposure, lymphocyte become activated and produce certain concentration of antibodies with low concentration, like here, okay, for initial exposure due to vaccination. What happen if the real pathogen enter our body? We will start second immune response, fast and rapid production of antibodies with high concentration due to presence of memory cells created or evolved during first immune response or primary immune response. Okay. Now, question number 4. Complete the sentence about the mammalian nervous system. Mammalian nervous system. The space nervous system consists of the brain and spinal cord. And the space nervous system consists of nerves outside the brain and spinal cord. So, here, look at the figure. We have two types of nervous system. The main nervous system, central nervous system, consists of brain and spinal cord, central nervous system, CNS, and the peripheral nervous system, PNS, nerves emerge from brain as cranial nerves and spinal nerves from spinal cord. Okay. Letter B. Figure 4.1 shows the parts of reflex arc. MNO, MNP, Q, R, ST for effector up to stimulus. Put the parts listed in figure 4.1 in the correct sentence and write the letters in the boxes. 2 have been done for you, where T and R, starting from T stimulus, and ending with R, response. Okay. How we arrange in correct sequence these letters. Look please to reflex arc. Reflex arc start with stimulus. Here stimulus. Okay, like pen, pinch of pen. Okay, heart of pen in our hand. What would happen? Sensation, okay, passes through what? Through from receptors in our skin, through sensory neuron, the sensory neuron, okay, to reach our spinal cord, okay. Here communicate with relay neuron, this one, then relay neuron communicate with motor neuron, where orders passes towards the effector, the muscle, to help our hand move away from the pen. Okay, that's called reflex arc. At the same time, when relay neuron passes orders through motor neuron to effector, the same message reach in another way towards the brain to tell the brain what has been happened due to puncture with this pen. Okay, that's called reflex arc. So most reflexes done by spinal cord, but little reflexes done by brain for memorizing the event that happened, okay, due to action of spinal cord. Okay, look please to the letters. 2 have been done for you. T stimulus, so passes the stimulus as sensation through sensory neuron. Okay, sensory neuron. Then before sensory neuron, receptor cell, here receptor cells, sensory neuron, so receptor cell first, so letter P. Okay, then sensory neuron, letter S, then letter Q, relay neuron. Okay, then N, N where N motor neuron, then R effector as muscle contracts to move away our hands away from the pen. Okay, the sequence represents the steps of reflex arc. Okay, letter I. State 2 stimuli that are detected by the skin. The skin as sense organ where a lot of sensation takes place by our skin. A lot of different sensory neuron types found in our skin. Okay, here what about 2 stimuli like touch, like temperature, coldness or hotness, like pain, like pressure. Okay, all of this are stimuli detected by our skin. Okay. Letter triple I, the pupil reflex controls the amount of light that enters the eye. See the name of an effector in the pupil reflex. Effector. The effector in our body may be muscles or glands. Muscle to contract and relax, glands to secrete certain chemical like hormones. Look please. See the name of an effector in the pupil reflex. Circular and radial muscles of the iris. Both, okay, manage or control the amount of light enter our eyes. Look here at intense light, what would happen to eye pupil? Become narrow due action of what? Both muscles. They are antagonistic muscles. They behave in opposite manner. One contracts, the other relaxes at the same time. Here during what? During intense light, circular muscle contracts, radial muscle relaxes. Okay, radial muscle relaxes. What about when subjected to dim light? Dim light, what would happen to eye pupil? Become more diameter, much wider. Why? Because the reverse, circular muscle relax and the radial muscles contracts. Okay, here the eye in bright light, like in this one, okay, circular muscle contract and radial muscle relax. Iris becomes wider. Here, where is the iris? The color part of our eye, all the diameter becomes wider, while the eye pupil becomes shorter. The reverse, pupil is smaller in diameter. So take care when we deal with iris and deal with pupil. Pupil is just the opening where light passes through. The iris means the colored part of our eye. Colored part. Okay. Now describe and explain how impulses are only passed in one direction from one neuron to the next. Okay, here we discuss something called synapses. Synapses. Here this part presynaptic neuron and with button swelling called button, okay, communicate with certain tiny space with another neuron, this postsynaptic neuron. Okay, here presynaptic neuron, postsynaptic neuron, in between synaptic cleft or gap. Presynaptic membrane, postsynaptic membrane. In the bottom here, a lot of mitochondria, a lot of vesicles containing neurotransmitter. Okay, we will take this box only and magnify. Look, what about these vesicles? This vesicle containing neurotransmitters molecules, where this vesicle move and diffuse with the presynaptic membrane and make something called exocytosis to release its content of hormone of neurotransmitter molecules, to reach this gap by diffusion, to reach receptors lie in the postsynaptic membrane. It allows the passage of nerve impulse in the next neuron. Okay, here return to the question, how the structure, how the impulses are only passed in one direction from one neuron to the next. We find certain important hints here by electron microscope. This part where vesicles found, here second part in between the two tiny lines, this is the gap or synaptic cleft. Okay, what happened? All synaptic vesicles lie in presynaptic neuron. Okay, all receptors lie and found on both synaptic membrane. This ensure one direction flow of nerve impulses from presynaptic neuron to postsynaptic neuron. Why? Because all vesicles here lie in one direction and all receptors in the second direction. Okay, so the answer, only the first neuron releases neurotransmitters through neurotransmitter vesicles. Only the second neuron has receptors for neurotransmitters. This ensure one-way direction. What's the rest of the answers? Look, neurotransmitter is released from vesicles into the synaptic gap, diffuses by diffusion. Neurotransmitter binds to receptors on the next neuron. Then this triggers a new nerve impulse in the next neuron. Number 5, the gene for red-green color vision is on the X chromosome. I mean, this example represents sex-linked trait. Sex-linked trait. There are two alleles for this gene. The allele for normal vision, color vision, is represented by the letter P capital. The allele for red-green color blindness is represented by the letter P small. Okay, the figure 5.1 shows pedigree chart for family in which some of the members are red-green color blind. Okay, squares for male. Okay, a circle for female, a plain square for normal color vision, shaded square for red-green color blind male. Okay, here also the same for female. Female is circle, white or non-shaded circle for female with normal color vision, shaded with red-green color blind. Explain why all the male children of parent 1 and 2 are red-green color blind. Where these children, male children, where number 7, number 9 are red-green color blind. Why? Look at their parents. Here their mother as it shaded circle, it represent what? Homozygous color blind. Their mother is color blind, while must the husband or the male here, the father only passes Y chromosome to his sons, so Y is not responsible for transmission of this inherited disease. Okay, so the answer now, why children, male children from parents 1 and 2 are red-green color blind. Number 1, parent 2, the mother is homozygous recessive. It carry XP small and passes XP small to all sons through the ova. Okay, while males only need both 7 and 9 male children need only one X chromosome from mother to be affected. Okay, while due to red-green color blindness is sex-linked disorder. Y is not responsible for this disorder. It's okay, Y chromosome is not responsible. Now for letter P, this problem, parent 4 is female who has normal color vision and is heterozygous. I mean XP capital, XP small. Okay, sorry. Complete the genetic diagram to determine the probability that the offspring of parent 3 and parent 4 would be red-green color blind. Parent 3 and 4. Look at 3 and 4 again. 3 and 4. Where is 3? 3, this one, and 4. Okay, 3 male affected by color blindness. 4 is heterozygous. Heterozygous can distinguish between red and green, but it carry recessive allele in its genetic genotype. Look here for 4. Now red green color blind male, I mean XP small y, and female with normal color vision but heterozygous for the trait, so it carry only XP small in its genotype. So its structure XP capital, XP small. This parental genotypes. What about gametes due to meiosis? Each gametes split, or each chromosome separated from other in meiosis. So XP small in one sperm, and Y, okay, doesn't carry any alleles, okay, and next type or second type of sperms. Here the ova with 2 types, XP capital, XP small. Okay, what's the problem now? The rest of problem, complete this table. Offspring genotype, how these gametes fuse together to form offspring. 2 types of gametes times 2 types of gametes, so 2 times 2 equal 4. First, second, third, fourth. Okay, this offspring genotypes. Number 1, number 2, number 1, number 2, 1 with 1, 1 with 1. Okay, 1 with 2, 1 with 2. Okay, there's 2 possibilities, 2 probabilities for sperm XP small fertilize the egg XP capital or XP small. 2 options, 2 possibilities. While sperm carry Y has also 2 possibilities to fertilize this egg or fertilize another egg. 2 possibilities. So 2 times 2 equal 4 individuals. Look now, 1 with 1 to form XP capital, XP small. Okay, so XP capital, XP small. It is recommended to start with capital first, then small. XP capital, XP small. Right. Number 2, XP small, XP small. Now we finished this options for this sperm. The second sperm Y, that responsible for male offspring, male child. XP capital Y, XP small y. Now look, offspring phenotypes, the features of all 4 individuals. The first one, female, but normal, carrier, heterozygous. Okay, here normal female, carrier. XP small, color blind female. Here normal male, XP capital Y. Okay, here XP small y, color blind male, From where this XP small in this color blind? From his mother. His mother responsible for transmission of genetic inherited trait that carried by X chromosome. Y is not responsible for this character. Now he asked me about probability of a spring having red-green color blindness. What is the probability? How many having red-green color blindness? How many out of 4? Here color blind. 2 out of 4. So 50% of spring carry this disorder. 50%. Okay. Now we reach last question about penicillin can be produced in fermenters. See the name of the type of organism that produces penicillin. Okay, name the type of organism. Penicillin from penicillium, from fungus, yes, or fungi, like this one, this penicillium. Fungus or fungi, accept penicillium, can be accepted in your answer. Okay, look please. Figure 6.1 shows the change in biomass. Biomass means the mass of what? Of organism that produces penicillin, the mass of penicillium fungus inside fermenter when grown in fermenter under controlled conditions. Letter X, Y, and Z. For x-axis, time the organisms are in the fermenter per hours, how many hours passed, and the biomass per gram per liter or decimeter cubic or liter. Okay, look please. State the name of the growth stages of the organism shown by the letters X, Y, and Z. This graph in this shape called sigmoid graph. Sigmoid graph. Ideal sigmoid graph. What about these letters? Look. The first few hours when we supply the fermenter with quantity of fungus, of penicillium filaments inside the fermenter, it passes stage called lag phase, where the penicillium as organism accommodate with the new conditions inside the fermenter. X, lag phase, period of slow growth, while cells adjust to new conditions. Okay, start to adjust to new conditions inside fermenter. Lag phase, growth is slow at this phase, just reach about less than or about, say for example, at after 20 hours it reach about say 8 gram per liter inside the fermenter. Okay, that's first stage, lag. While Y, logarithmic or log phase, exponential phase, rapid growth, steep growth, rapid cell division and population growth. Why? Because the penicillium cells as multicellular find all the nutrients, all its requirements of oxygen, of nutrients like glucose, like source of nitrogen, okay, to form new cells and start to division in rapid rate, okay, by mitosis. While Z is stationary. When the fungus increase in its rate of reproduction or rate of division, it reaches the stationary phase. Why? Growth rate slows as nutrients deplete and waste mark accumulates. Depletion of nutrients due to accumulation, large population number of penicillium cells. All consume the same nutrients, all compete of the source of oxygen and source of nitrogen, all in the same container as the fermenter, all compete to these resources. So also due to metabolism, due to respiration, waste accumulate inside fermenter. This leads to slow growth rate, okay, the stationary phase. Now this the design diagram of fermenter containing organisms that produce penicillin. Okay, explain why air is supplied to the fermenter shown in figure 6.2. Air filter to become sterile without microorganisms to avoid competition with the main organism penicillium inside fermenter. Here, why air is supplied? Oh sure, this fungus is aerobic in respiration. Okay, it carry out aerobic respiration to release its energy needed for growth and metabolism. So the answer, organism need oxygen for aerobic respiration. Okay, during aerobic respiration it releases energy for growth and metabolism. Okay, what's the function of bottle? This extra question to mix content, okay, and the nutrients with the fungus, and prevent accumulation and the precipitation of fungus mycelium and hyphae down at the bottom of the fermenter to make them homogeneous with the content. Look, explain why the temperature in the fermenter must be controlled. As you know, temperature affect metabolism, affect enzyme action, affect the 3D structure of the enzyme and its active site. Okay, look. First part, respiration produces heat, which can increase the temperature. So sensor or probe, okay, can sense any change inside the fermenter and connect this probe with data logger or computer to mention or to record the temperature each time during working out of the fermenter. Respiration produces heat, which increases the temperature. Enzymes are involved in microbial metabolism and can become denatured if the temperature is too high. Okay, look at the denature of enzyme. Here proper or this good enzyme structure, here bad, where denaturation takes place due to heat exposure more than optimum. Okay, now this only protein no longer act as enzyme, only act as protein like any protein, but no longer act as enzyme. It lose its action, its job. Okay, enzymes are involved in microbial metabolism and they can become denatured if the temperature is too high. Maintaining optimum temperature ensures maximum yield of penicillin. Of sure, where penicillium uses enzymes to produce its metabolite, I mean penicillin. Okay, see the role of bubbles in the fermenter. I answer this question before. Okay, again, see the rule of bubbles: mixing, stirring to keep conditions uniform, conditions of pH, condition of temperature, homogeneous distribution of nutrients throughout the fermenter, top and the bottom and in between. Okay, ensure even distribution of oxygen, nutrients, fungi, and temperature throughout the fermenter. Okay, other than penicillin, state the names of two commercial products that can be made using fermenters. Two commercial products can be made using fermenters. We can produce insulin in large scale used for diabetes treatment, okay, especially diabetes type 1. Okay, also here we oblige and afford all conditions needed for bacteria, okay, to grow inside fermenter, okay, and produce to produce insulin, where we treat this bacterium with plasmid, recombinant plasmid containing human insulin gene, okay, and the bacteria obliged and can produce a lot of insulin hormone inside fermenter. Okay, insulin is harvested, purified, and then taken as treatment for treat diabetes. Okay, what about another name of commercial products? Mycoprotein. Myco means fungus, like trademark coron used as meat substitute. Look, this from fungus, from types of mushrooms. Okay, its taste and its structure mainly like meat. Look at this texture and its shape like meat. It's used in large scale. Okay, this second commercial products can be made by using fermenters. Okay, coron. Thank you for watching.